So Céline, what can you tell me about this case?
It's a suicide case, a male, 37 years old.
With the contents of this box, you'll be able to get inside the head of a suicide victim.
How did that person die?
It's a gunshot wound. We need to still interview the family to get more information.
Najib is director of the Quebec Brain Bank,
Okay, here it is.
where they receive 80 to 100 human brains every year.
Let's see what we've got. Okay, so it is a nice specimen. It's a tremendous asset for research in neuroscience and psychiatry to understand how the brain works, but especially how brain diseases occur. We really need to analyze human brain tissues. There are some pretty good animal models for different disorders, like Alzheimer's disease, for instance, but they're only models, and we really need to study the real thing.
The real thing weighs about 3 pounds, 1,400 grams or so on average, and looks like a uniform mass of folded, crinkled flesh. And yet every small piece of it plays a different role in driving the human machine.
You can see here we have the right hemisphere of the brain, the left hemisphere. This is the front of the brain, where you have in particular the prefrontal cortex, the back of the brain with the occipital lobe. So we have the frontal lobe, occipital lobe, we have the temporal lobe here. Okay, and when we flip the brain over, we see the cerebellum here, and we can also notice the brainstem, which is located above the cerebellum.
All in all, they have about 2,500 brains here.
As you can see here, we have several samples which are preserved in formaldehyde solution. So you have an example here where you see the hemisphere in formaldehyde, and the S here means that it's a suicide subject. But of course we have hundreds of other subjects that are from Alzheimer's disease patients, people with senile dementia, Parkinson's, and other neurodegenerative disorders, and of course also of controlled subjects, so healthy subjects without any neurological affliction. And what we have here are minus 80 degrees freezers, where we keep the other half of the brain. As you can see, there are several samples in each freezer, so we have about 150 brains in this freezer in particular. And so they're kept at minus 80, which prevents protein degradation. And so that's a great way to preserve the brains. This is one of the samples. You see the whole hemisphere has been cut into thick sections, which allows us to dissect specific regions more easily afterwards.
It's hard to imagine wanting the job of slicing and dicing, but Ruth gets right in there.
I am making sure that both hemispheres are well separated, so that when I actually cut through the corpus callosum, I do not cut any of the cortices.
Every donated brain is split in two. Each hemisphere is weighed.
657.
One half is preserved in formaldehyde, the other sliced into smaller sections for freezing. The brain bank gets many donations, but not always the kind they want.
We can never have too many brains. However, we do have a problem which is encountered by all brain banks across the world, is that we have fewer control subjects, let's say, that donate their brain. When I say control, I mean people without neurological disorders that died from car accidents, for instance, that are essential for research because we need to compare the results obtained in diseased brain to normal, healthy brains.
Comparisons like those are the clues researchers need to decipher the mysteries of the brain.
Well, this is a subcortical region, so we're in the midbrain here. And what's interesting here is that you see this, this dark region on both sides of the brain. And this is called the substantia nigra. These cells are the ones that degenerate in Parkinson's disease. So we have a high demand for this brain region.
And by comparing control brains with those of suicide victims and people with mood disorders, Najib's team discovered something about what's called dendritic arborization.
If you think about these neurons, which are called pyramidal neurons, they have a pyramidal shaped cell body. They project what we call a dendritic tree, and it really looks like a tree, in fact. There are several branches, and these are sites of contact with other cells. Okay, so it's very important for cellular communication in the brain. What was found recently is that by analyzing these different branches, there's a reduction in the number of a specific type of branch in mood disorders compared to control subjects.
They don't know exactly what the fewer branches mean yet, but Najib is dedicated to finding a way to help people with mood disorders and potentially save lives.
What's really appealing about the brain is that it's an extremely complex organ, and we know very little about it. And in fact, the brain is who we are, is what we are. And so by studying the brain, we kind of peer into human nature.
Don't go away, because after the break, you're going to learn about